Welcome to the HIDS Virtual North America Regional Session. We hope you're enjoying the conference. My name is Shereen Darwish. I'm the North America Market Development Manager at Thermo Fisher Scientific, and we're delighted to bring you this next session featuring 3 expert presentations focused on how DNA is used by law enforcement and crime labs to solve crime. Please be sure to submit your questions for each of the 3 presenters in the Q&A box on your left. And as a reminder, this session offers free continuing education credits. Click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. For a complete biography on all 3 of our speakers, please visit the tab at the top of your screen. It's my pleasure to introduce to you our first presenter, Stephanie Regan, Crime Scene and Laboratory Unit Supervisor at the Kauai Police Department.
Thank you for attending HIDS, and also thank you for attending this presentation block. My name is Stephanie Regan, and I'm the Supervisor of the Crime Scene and Laboratory Unit at the Kauai Police Department. Today I'll be speaking on our use of rapid DNA for a helicopter case for disaster victim identification. So just a quick disclaimer, this is an actual Kauai Police Department case, and this is not for any legal or prosecution purposes. Out of confidentiality for the victims and those affected by the crash, do not capture or share any part of this presentation except with the express written consent of the Kauai Police Department. Just a quick content warning, the following presentation does provide an account of a disaster victim identification from an actual helicopter crash, so images and content may be disturbing to some viewers. Therefore, viewer discretion is advised. I want to take a brief moment just to pause and acknowledge those that were lost during this crash and the families that lost loved ones during this incident. On December 26, 2019, we had a crash on the northwest coast of Kauai. Kauai is the westmost island of the Hawaiian chain. And here's a general depiction of the start of that cliffside. So the helicopter went down generally in this region. And to give you a little understanding of how tough this terrain is, this is how I ended up having to get into the scene to actually do any victim identification recovery and helicopter part recovery. And so basically it's just steep cliff all the way throughout this. And the helicopter impacted. We'll show a quick video here next, but the helicopter actually impacted one of the cliff sides, where it fell approximately 100 feet to the bottom and caught on fire there at the bottom, which further complicated all of the recovery of the remains as well as the reconstruction of the actual crash. So you'll see in the video, it's actually myself and one of our detectives there at the bottom of the cliffside, actually in the cockpit area, and we're doing some parts identifications and photographies for the engineer. But you can actually see how steep that cliff is behind me. And then the arc at the top of the dirt is actually where you will have, you'll actually see where the actual aircraft impacted. To give you a quick overview of the actual timeline of the victim identification process, the helicopter crash actually happened on the 26th. It was not found on the 26th, and it was actually a missing aircraft during that time. So the helicopter crashed on the 26th and was actually located on the 27th, where remains recovery was able to be taking place. So based on how difficult the terrain was, the recovery actually spanned over most of the weekend. Our medical examiner is actually off island, So they have to fly over to do autopsies. So it actually took a few days for them to also get here, and started the autopsies on Tuesday, the 31st. Most of the autopsy took place on the 31st and the 2nd, where during that time, 28 different body parts were able to be identified. At that time, 16 were determined by the medical examiner to possibly be viable for DNA analysis. They did take pictures of the 12 additional samples, but at that time they didn't sample them. I began my testing on the 2nd and kind of pretty much all the way through till the 8th. On the 3rd, I actually sent those pictures out to my support team at Thermo Fisher Scientific of the 12 remaining samples that were deemed originally as probably not being able to be identified. And from case experience, they actually told me that it would be useful, or it would be potentially useful, to go ahead and actually test those samples. So Monday, I ended up sending a colleague back to take additional samples of those 12 samples that weren't originally taken, and we ended up testing those as well. One of the biggest things in this case was during the autopsy portion, they were only able to identify 6 different distinct bodies from doing body part elimination. So from the offset, we were a little bit concerned that we wouldn't be able to ID all 7 victims, and also weren't, in that regards, going to be able to get the death certificates. And anyone that's done disaster victim identification knows if you can't get the death certificates, legally it becomes very challenging, and it puts a huge burden on the families on trying to move forward with all of the different processes needed after end-of-life care. So, from the offset, we weren't sure we were going to be able to do that. And within 3 days from the time that we finished the autopsies and had the samples to test till Sunday, and again, running pretty much even half capacity on the weekends, we were able to ID all 7 victims via DNA by the 5th on Sunday. So that was huge, knowing that we would be able to cut all 7 death certificates to allow that legal process to move forward. Again, on Monday, those 12 additional samples were taken, and on Monday, and then on the 7th, I was able to process all 12 of those samples, successfully IDing every single one back to the person that they came from. And then on the 8th, approximately 2 weeks from the crash, we were able to authorize release of every single one of the 28 samples. The system at the bottom, the Applied Biosystems Rapid Hit ID system from Thermo Fisher Scientific, is what we ended up using to ID all of the different remains. So a quick overview of the cost savings that we had in this case, as well as the time savings. So based on the in-house rapid DNA processing, we only spent about $3,200 to process all of the different samples that we needed, both family samples and the remaining samples. And we were able to release those, as you saw on the last slide, on January 8th. For us, we didn't think that our state lab would be able to take the identification process for this case, so we would have had to go to our outsourcing private lab. And our private lab is actually on the mainland, so that adds a little bit of time to our actual turnaround time for identification. So it takes a few days for us usually to get the authorization and paperwork and fiscal stuff done, and then as well as shipping time to get those samples out. So as you know, most of the companies have different outsourcing ones. So for us, the fastest we would have been able to do is a 5-day expedite on the DNA processing, and about 10 times the cost. So it would have been $32,000 in comparison to $3,200. So about 10 times the cost, and we still wouldn't have been able to release the bodies for another few weeks. And then, you have different outsourcing expedites, so it would have ranged in price a little bit, but still almost 10 times as much cost, and we've been looking for at least another month based on the different expedite charges until we would be able to release those samples. Our standard outsourcing time still would have been 5 times the cost of our disposable cost, and it would have been about 2 months later that we would have been releasing those bodies back to their families. So just a quick highlight summary. All 28 body parts were able to be identified and authorized for release within 2 weeks of the crash, and actually 1 week from the autopsies being completed and testing being able to be started. All 7 victims were identified via rapid DNA within 3 days of the autopsy completion, and those were weekend days as well. For this one case alone, we were able to use rapid hit ID to save between 20,000 to 30,000 and up to 2 months of time. And again, that's being able to release remains back to their loved ones and let them move on in the process of having closure. Okay, so lessons that we learned throughout this process, first and foremost was that with the burnt remains was to test everything. So again, initially we didn't take samples from all of the more burnt remains. And this is a good example of some of the remains that we had. So as you can see, there is a lot of fire damage to the smaller remains. And we were able to end up going back and identify every single one that was determined to be human. So at the end of the day, if you can determine that it's actually a human tissue, go ahead and actually test it. This is the sample that actually was the worst profile that I was able to do. But again, this is very fire damaged and only took a very small sample from this tissue, and we're able to get enough of the profile to be able to identify back. Again, if you're doing a large database search or anything like that, probably wouldn't be able to make the ID, but with the N of 7, it was able to give enough profile to be able to attribute that back to the person that it came from. Here is a muscle tissue, and again, very badly burnt. And for a little bit of a perspective here, this bottom of the tube here is only about 8 millimeters wide. So you only need a very small sample to be able to actually run the rapid DNA system on it. This sample is actually pretty good. You can see that the small sample that we were able to take for actual testing is pretty viable tissue. But in this circumstance, you had most of the end of this bone and also the rest that wasn't sampled that is pretty badly burnt. And again, this is only a really small sample, and it's only about as thin as the scalpel itself. Here's an example of brain tissue that was found. And again, it's covered in a lot of dirt and as well as burnt sample. And if you see here in that top left portion or top center portion of the sample, we were able to take the swab and get enough clean sample. All you need is a very small amount on the tip of the swab to be able to load into the rapid hit cartridge and run, and I think this one got actually a full profile. Another lesson learned was that with burnt remains comes ash, and with ash comes sediment. So this is actually the first burnt muscle that I ran, and you can actually see that if you look in the well, you can see the ash kind of building up in the well, as well as running down into the capillaries of the rapid hit cartridge. So you can see the ash going down into both the that first well, as well as both of the capillaries right there in connecting the cartridge. This one actually caused a ladder error, but we were able to recover that in the data analysis phase. We did rerun that sample just to be sure, but it was actually a recoverable error that occurred. To resolve that moving forward, we did a very simple wash. So nothing fancy, but we just took an Eppendorf tube, placed our sample inside of it, and then did a quick wash with sterile water. Turned it up and down a few times, and that was it. So we usually did a double wash. This helped in both getting rid of that sediment and also kind of doing a pre-hydration of the sample. And we didn't see any errors throughout the remainder of the testing after that point. The last lesson learned is that burnt remains is going to be degradation, and the samples and the results and the data that you're going to get is going to reflect that degradation. So with that, a lot of people are thinking on the rapid DNA side that it's a hands-off system. But the hands-off system portion of rapid DNA is really designed for blood and buccal samples. So with that, with the degradation samples, you will have to have somebody that can do the data analysis and data review. So you'll need somebody to be able to go in the trace files and actually confirm whether or not certain calls are correct and clean up that data. So then you can go back and do your match comparisons or the kinship analysis. Further, the Rapid Hit ID software is really, again, designed for the hands-off blood and buccal type comparisons. So you might have to do some troubleshooting with these degraded samples as far as being able to actually generate those reports if you're relying on the Rapid Hit software. So I know a lot of labs, they do use both the built-in software, or they might have secondary software that you're doing the analytics part of it in. So if you're doing that secondary analytics, no issue there. But if you're trying to use the Rapid Hit software, you may have to do some troubleshooting. But in this, I was in direct contact with the Thermo Fisher scientific support team, and they were excellent in giving me timely turnaround and working through some of those issues on the back end so that we could actually generate all of the match reports needed, as well as the kinship analysis that was done. So just some things to know. Again, we were able to work through all of those errors, and luckily enough, we do have the biological backgrounds in-house for us to be able to do all of the data analysis. With that, I'll take a few minutes for questions. My contact information is here on the slide. So if you have any questions post-symposium, then I'm happy to answer any emails or calls that you may have. Thank you.
Well, thank you, Shereen, and thank you very much for this opportunity to present to you virtually today. I really appreciate it, and I'm thankful for the platform to be able to do this from. And today what I want to talk to you about is maintaining productive momentum in a rapidly evolving DNA lab. This is a reality for my laboratory right now, as we've spent the last several months becoming much more productive in the work that we do. And it's one thing to be able to become more productive and change what needs to be changed to get there, but it's quite another issue to maintain that productivity, because there's various factors involved in how you move from where you were to where you want to be and keeping that going in the right direction. So if you'll indulge me for just a few minutes today, I would love to just share with you my thoughts and experiences from our team's perspective as to how we are doing and getting towards where we eventually want to be. Now, this is something that nobody wants to hear from any biologist. That's the facts of life, but I'm going to share them with you anyway, especially when it comes to the facts regarding having to change, because some of it is hard. Change is really hard when it comes down to implementing it. Change is also fairly uncomfortable for the most part, because we get pretty comfortable in our routines, in our processes. We get comfortable with the people that we work with. And so to change all of that is quite uncomfortable. And it's also exhausting. Change at first can be exciting, but it's almost like an adrenaline kick where you start to do something and everybody gets excited and can see the big picture. But after you've been going for a little while and that initial excitement is worn off, you suddenly get exhausted. And it's really important in the middle of all of this that you can still maintain momentum. Change is also risky. So going into any kind of full-scale change, especially within a laboratory that's doing very important work that affects people's lives on a daily basis, you have to weigh up the risk. Is changing this process, or is changing this instrument, or is changing this chemistry, or is even changing to a different workflow and different personnel being involved, is it worth the risk? And so that is something that is a fact of implementing change so that you can continue to evolve as a DNA laboratory. And in forensics, of course, change is slow. So back to that first point of implementing because of excitement and knowing that you want to see the end product of the big picture. In forensics, we all know that that now takes a long time for some good reasons. Of course, we deal in a legal system that has to be very careful, and rightly so, that everything is tested and validated and tested again and changed and tweaked and trained, and everything involved with it takes time. And so just by expressing the fact that you want to move to a better way of doing things, a more productive way of doing things, doesn't necessarily mean that that idea is going to be implemented anytime soon. So in the maintenance of the momentum, especially within the arena of forensics, it can be quite frustrating to implement change because we know ahead of time that it's going to take a long time to get to where you want to be. But on the upside, change is certainly necessary. We're a scientific field, and we need to move with the science. We need to move with the technology. We need to be able to offer to our community, our customers, the people we serve on a daily basis, we need to offer them the latest and the greatest as is appropriate. So we can't stay back in the ways that we were doing things 5, 10, 15, sometimes 20 years ago. Change is absolutely necessary so that we can give the best possible work product to those people that we serve every day. And change can and should be productive. And if it's not, then you start again or you scrap it. But that's part of the risk that you've assessed before you've gone into this endeavor. So for us, what has changed? In reality, we have become fully staffed over the last couple of years, which is something that I know many labs struggle with. But for ourselves, one of the big changes that needed to be made was more personnel. And so I'm thrilled, as are our management team, that we have the people in place at the appropriate level to do the job that we want to do. We've changed our training program, which came with its pros and its cons, but instead of doing things a certain way and waiting for everybody to train in the entire process, we've gone to a task-based training program where we can introduce our analysts, especially the new folks who've just come on board, into the actual workflow at a necessary stage so that we can, one, make them feel productive, but two, and more importantly, improve our workflow with their input into these key areas. Because of that, and because of our instrumentation and our upgrades through technologies and validations, we've increased our throughput capacity in the last 10 to 15 months by 500%. That doesn't mean that that's what we're seeing yet, but we've given ourselves that room to grow. And that is really encouraging for us, sometimes a little scary, but it's certainly encouraging to know that that's what we can fill. That's kind of the shoes we can step into. And as we continue to get better and maintain this improvement and productivity, we know what it is that we can reach as a ceiling because of our increased capacity. In the last 10 months, for example, and I'll show this figure again in a few moments, we've increased our productivity 100%. So you'll see the direction and the momentum that we've got going on in our lab right now. And I'm thrilled that this year, just a couple of months ago in April, for the first time ever since I arrived in this lab, which was a little more than 3 years ago now, we've completed more cases than we've had requests to perform work and analysis on. So I'm happy to see that. And again, it's just another great indication that is shared with our team, our management, and our wider public community to show them that we're doing what they're asking us to do and we're doing it well. We also set realistic but ambitious goals. That's changed because the old habit really, and I'm sure this is the same in multiple locations, is to just keep plodding along in that comfort zone. But in order to get better and to improve and to increase that momentum and productivity, you have to set some ambitious but realistic goals. And we've done that together, and I'd encourage you to do exactly the same. Don't just be happy, even if you're a highly productive lab that's getting the work done in a timely manner, still go ahead and set ambitious goals that are realistic and continue to improve, because losing that momentum eventually through exhaustion can lead to some kind of backtracking. And so for us, we set those goals that were realistic, but they were really ambitious. And then what we did in order to hold our feet to the fire was to publicize our expectations. So on local news channels, through public meetings, certainly to the command staff in our agency, all of our expectations and goals were publicized. And that makes me very nervous, but that's okay. That's why I have the role I have, and that's why my manager has their role too, so that we can continue to push that expectation downwards while also being accountable upwards for what it is that we really want to do. So here's what I was talking about a little while ago, just a really quick comparison of the 10 months in blue prior to implementing all of our changes in the lab. So the y-axis is just the percentage of cases cumulatively that were put out over that 10-month period. And so, of course, month 10 is 100% of what we did for the prior 10 months. And then comparing that to the 10 months directly after implementing the changes that we've done so far, we see an almost 200% realization of what we'd seen in the previous 10 months there. So with the curve still trending upwards for us, we're thrilled to see that we're going in the right direction. And for me, to know that this just involves 20% of the new personnel being productive in these figures gives me great hope that that will continue to rise and rise and rise over the next year to 2 years, and will certainly be in a wonderful position in the very near future. One of the things that really is exciting for me is to be able to change the instrumentation and the technology. Is just a small number of the new instruments that we were able to bring on board during this period of change, from automated extraction robots to upgraded CE instruments and more consistent PCR instrumentation, and even a rapid DNA, the rapid hit ID DNA system for our CODIS confirmation samples. These all together, along with the new protocols, the new validations, the new chemistries, and certainly by no means least the new staffing, has caused us to have these increases in productivity and maintain that momentum. So why has the change been effective? Well, what we did first of all was identify our problems. And our problems were really with productivity. We were asked to do a job and we weren't getting it done in a timely manner. We were getting it done eventually, but we needed to get it done better, more efficiently, and more effectively. We set ourselves some very clear goals, timelines, dates, numbers that we had to achieve. And I would encourage you to do the same. Don't be fluffy and fuzzy about the things that you have to achieve. Be very clear and succinct in identifying what it is that you need and want to do. We garnished strong support, and that support was both from our leadership within my agency and the community at large. Again, that came with the publicizing of our expectations and our challenges too. But getting the leadership and the community on board with us and wanting to have us succeed was really important in obtaining their support. We, by no means, in a small way, partnershiped with Thermo Fisher Scientific for our validation studies, our upgraded chemistries, our new instrumentation, and I couldn't be more grateful to them for their help, their encouragement, and their constant and consistent crucial support over this time period. And we have big plans for the future too, where we're continuing to upgrade our workflows and our systems too. So that was certainly a huge part of our success. We had in-house commitment from those in the same level as myself and the bench level analysts too. And it's really important that as a management team, we're able to maintain enthusiasm and the resolution to succeed from the analysts on the ground who are actually implementing this change and performing the work that needs to be done. So finally, what does that mean? Well, for us to maintain this momentum, we have to say, what are our minimum expectations? What's the projections that we have in order to keep this going in the right direction? And here it is, and it might just look like numbers to you, but for me, these are realities. And so in the remainder of this current calendar year that we're in, we've predicted that at minimum we're able to complete 152% of everything that we know because of the historical data we've gathered of all that we're going to be asked to do in this remaining portion of 2020. So that's great, and that is something that we have as a very clear goal to achieve. But then we needed to look ahead to 2021, and as we carry over the current backlog to next year as well, a portion of it at least, and expect to get the regular number of cases in-house for us to complete, we predict at minimum we will cover everything that we're asked to do, including that backlog, and by a little bit more. And again, I say that's at minimum. I do expect more, but I want to be realistic about what we expect. And if you're not realistic, then you're not going to maintain that momentum in progressing through the work that you need to do. So the result of that is very clear in my mind. And the result is that we're going to completely eliminate our backlog, and we're going to complete all of our in-house cases within our state-mandated time frame by the end of this year, which thrills me, and not only me, but also my leadership team too. In thinking about next year, it also means that we're going to have a surplus capacity available to us in 2021, which means that we can do even more, maybe decrease the turnaround times more, we can accept more case types of cases that we don't currently accept in our laboratory, and look ahead to other validation studies, research, and special projects that we have in our mind and in our hearts to get on with. So that's what we can look forward to because of this. And with that, I just want to thank you and hand it back to Shereen for any questions that might have come in during this presentation. Thank you very much.
I'm going to give is around talking about new tools that we have in our portfolio to improve efficiency and save time. Namely, we're going to focus on the 3500 data collection version 4.0.1 software and the SeqStudio data collection version 1.2.1 software. So jumping in, I'm going to start talking about a new pull-up reduction algorithm that is included in both of the software versions that I just mentioned. So I'll give a little bit of theory about the algorithms, and they're slightly different between the two software platforms. And then I'm going to run through an STR kit comparison where we took different STR kits, both from Thermo Fisher and other companies, ran a similar data set, and looked at the benefits of pull-up reduction with the AB chemistry on both software platforms. So to start, a quick overview of pull-up, just so that everybody is on the same page. We generally expect to see pull-up that is around 1 to 3% of the parent peaks. And we see pull-up in one instance because calibration, the matrix spectral calibration isn't accurately applied to the data. We know with off-scale data, this ability to accurately apply the calibration can be even more discrepant, and you may see higher pull-up peaks in off-scale data outside of the 1 to 3% range. Typically, the pull-up that we see can be broken up into two different categories. The first is called spectral pull-up, and the reason you see spectral pull-up is because the calibration dye data that you run when you do a spectral calibration doesn't perfectly match up to the sample dye data. And the reason for that is when you're doing a spectral calibration, a manual calibration, you're using pure dyes. And a matrix is generated and those values are applied to your data to deconvolute dye overlap. But in your sample, the dyes are attached to DNA fragments, and that could affect the spectra slightly. So you're not applying a correction that is exactly apples to apples. The difference between the matrix you see with pure dyes and what's actually happening in your sample leads to spectral pull-up. This type of pull-up in the analyzed data is centered under a parent peak, and you can also visualize it that way in the raw data. The second type of pull-up is instrument-specific pull-up, and this all has to do with the way the fluorescent molecules are moving in the capillary and specifically across the detection cell. So when the molecules enter the detection window, center within the window, and exit the window, it causes the spectrum on the CCD to shift, and you can see instrument-specific pull-up. In the analyzed data, this type of pull-up could be slightly shifted from the center of the parent peak, and in the raw data, it takes on a sinusoidal shape. If you have both type of pull-ups occurring in the same dye channel at the same time, they can be additive. So you could see an increased percentage at that location of the pull-up peak. So when we came out with data collection version 4, we discussed the introduction of a new pull-up reduction algorithm. So with the introduction of the 3500 HID data collection version 4.0.1, we introduced a pull-up reduction algorithm. And this pull-up reduction algorithm is going to utilize sample-specific spectral data for AB dye sets, G5, J6, and J6T, to generate a matrix that's more appropriate to the sample that's been injected. If for some reason you have a sample with limited data, like a low-level sample where there may only be a couple peaks, the algorithm is going to say, hold on a minute, there's not enough sample-specific spectral information here. Let me revert back to the manual calibration and apply that to the data. So you're going to get a level of pull-up reduction either way. It's just whether it's the traditional pure dye calibration or if it's the added benefit of the pull-up reduction algorithm using that data from the sample itself. All pull-up reduction is taking place prior to the creation of the HID files, so when you view the raw data in IDX, the pull-up reduction will already be applied. In the SeqStudio HID data collection version 1.2, there's actually 2 levels of pull-up reduction. The first level, which is called auto-spectral or auto-calibration, is similar to that of data collection 4.0.1 on the 3500. Sample-specific data is being used rather than data from a manual calibration if there's enough quality data available in the sample injection. What's different is this is for any dye set. So whether you're using an AB chemistry or another chemistry, you will get this first level of pull-up reduction. Also, what's different is if there is a sample with limited sample-specific data, again, we'll go back to that example, a low-level sample with just 1 to 2 peaks being called. Rather than going back to the manual calibration first, if there has been a good auto-calibration or a good sample-specific calibration in that capillary with that dye set, that auto-calibration will be used. If there hasn't been, then you will go back to the manual calibration. The second level on the SeqStudio to help reduce pull-up is called marker-to-marker correction, and this level 2 is only for AB dye sets, again, G5, J6, J6T. This is an optimized correction factor for each marker in the kits. You select your kit during plate setup, and this is what enables this marker-to-marker correction level of pull-up reduction. On the SeqStudio, it's similar. All pull-up reduction is taking place prior to the creation of the FSA files, so your raw data in IDX will have the pull-up reduction applied. So let's jump into the kit comparison. A little bit on the general conditions of this study. We did all of our applications on the Applied Biosystems ProFlex system and all analysis in GeneMapper IDX 1.6. We utilized minimum thresholds, which I'm going to talk about a little bit more on the next slide, for all kits that were kit and instrument specific. As far as the parameters that we used for amplification and injection, also default DNA input, we used the different manufacturers' recommendations. So you can see there were 5 kits tested, 2 from Applied Biosystems, Global Filer IQC, and NGM Detect, 2 from Promega, Promega Fusion 6C, and Promega PowerPlex ESI 17 Fast, and a kit from QIAGEN, the Investigator 24-plex QS. You can see there's 2 different cycle numbers, either 29 or 30, and different DNA inputs, either 1 nanogram or 5 nanogram. Samples that we used, we wanted to use non-probative samples. That's what the NP stands for on this slide, samples that may be seen by a crime lab. So we had a swab from a cell phone, a cigarette butt, swab from a baseball hat, and blood on cotton. We also utilized a kit-positive control. We did this kit comparison on one 3500 XL and two SeqStudio instruments. So a little bit about the minimum thresholds and how we establish those. What we do is we take at least 7 NTCs from each kit and analyze it in IDX at 1 RFU. We remove clear artifacts like spikes and pull-up from the size standard, dye artifacts, things like that. Everything else that's left behind, we look at the average peak height and we add 10 standard deviations. This is called our limit of quantification, where you expect nearly all the noise to be below. We round that LOQ to the nearest 5 and get a minimum threshold. We did this, like I said, per kit, on the different instruments, and per dye channel. So for the 3500 data across the different kits and dye channels, we saw a range of 35 to 85 RFU. On the SeqStudio, the range is a little bigger, but we did have 2 instruments, so just keep that in mind. We saw a range of 25 to 140 RFU. And this way, when we were talking about pull-up artifacts, we made sure that we were using thresholds that were appropriate for the kit and instrument being tested. I just want to go over a little bit of terminology that I use in the results section before we get into that. I talk about traditional pull-up, and this is pull-up where it's very easy to calculate a pull-up percentage. So where a single, either spectral or instrument-specific pull-up peak falls clearly below a parent peak. I'm also going to talk about more complex spectral artifacts, and sometimes we see this as some people may call it pull down or a bridging effect in the data, and you can see that on the figure in the bottom. It's harder to calculate a percentage and identify sometimes which parent peak the pull-up is originating from. So for these type of artifacts, the complex artifacts, we counted the pull-up peaks if they were called an IDX, but did not calculate a percentage. And that should make sense as we start looking at results. So looking at the 3500 results, again, this was one 3500 XL instrument with 4 non-probative samples and 1 positive control. On the 3500 in data collection 4.0.1, just a reminder, AB kits will have pull-up reduction enabled, non-AB kits will have pull-up reduction disabled. You can see that this table is broken up into a left side and a right side. On the left side, we're talking about those traditional pull-up peaks where we could calculate a percentage. And on the far right column, the complex pull-up peaks, which is just a count, no percentage calculated. So a few things to pull out from the table. I also have a chart of this to see easier on the next slide, but you can see that the average pull-up across all 5 kits was below 3%. In the non-AB kits, there was some pull-up above 3%, and even a couple of peaks in 2 of the kits above 5% pull-up peaks. You can also, if you see the total number of peaks and the total number of complex peaks, the pull-up is greatly reduced for the Applied Biosystems kits. So just looking at it in a different view, if you look at the chart, what you see for the different colors is spelled out in the legend. The color red indicates complex pull-up. The lightest blue is pull-up greater than 5%. The next color, darker blue, is pull-up less than 5%. And the purple is pull-up that's below 3%. So you can see that visually here. The number in the gray boxes is the total number of pull-up peaks, traditional pull-up and complex. So the overall conclusion here is that the other kits, meaning the Promega kits and the QIAGEN kit, have at least 3 times the number of pull-up related artifacts compared to Global Filer IQC and NGM Detect. So you can see the significant decrease that the pull-up reduction algorithm has to offer. To consider this, if you were an analyst, you may say, okay, but what does that mean to me? We put together some numbers around time savings. So estimating it takes about 30 seconds per pull-up edit By the time you identify an artifact, go into the raw data, maybe have to do a calculation on percentage and make the edit. If we consider that the Applied Biosystems kits had 70 pull-up peaks in 5 samples, which is similar to what we saw in this data set, that would mean that the non-Applied Biosystems kit would have 3 times that, or 210 pull-up peaks in those same 5 samples. So just for editing pull-up, this would be a difference of taking either 35 minutes versus an hour and 45 minutes. And if we consider then that this time for review, we can double those numbers. So an hour and 10 minutes to just edit pull-up in 5 samples for an AB kit versus 3 hours and 30 minutes for the other kits. So looking at the same 4 non-probative samples and positive control run on the SeqStudio instruments. So again, there were 2 SeqStudio instruments. We have the results here. As a reminder, AB kits have both auto spectral and marker-to-marker correction enabled. The non-AB kits have auto spectral enabled. Again, you can see that the total number of pull-up peaks is greatly reduced for the Applied Biosystems kits. And again, the average percent of pull-up for that traditional pull-up is less than 3% of the parent peak across all the kits. You can also see that for the non-AB kits, we're again seeing pull-up outside of that 1 to 3% range, and even in some cases above 5%. To have a different visual look at this, you can see here a very similar chart to what we saw before, with the different colors representing either the percent pull-up or if it was a complex pull-up peak. In this case, there are kind of 2 conclusions drawn. For Global Filer IQC, the other kits had at least 13 times the amount of pull-up artifacts, compared to NGM Detect, about 4 times the amount of pull-up artifacts. So when we look at this for time savings, I broke it down into 2 different analyses here because I wanted to capture the difference between Global Filer IQC and NGM Detect. So again, considering 30 seconds per pull-up edit, if Global Filer IQC had 3 pull-up peaks in 5 samples, the other kits would have 13 times that, or 39 pull-up peaks in those samples. And with time for considering time for review, we're talking about 3 minutes versus 39 minutes to edit pull-up in those 5 samples and review those edits. For NGM Detect, if we say that there were 10 pull-up peaks in 5 samples and the other kits had 4 times that, or 40 pull-up peaks in 5 samples, we're talking about a 10-minute analysis and review time versus a 40-minute analysis and review time. So overall, the conclusions that were drawn here was that the 3500 data collection version 4.0.1 and the SeqStudio data collection version 1.2.1 can streamline data analysis, especially when used in conjunction with AB-STR kits. So we saw anywhere from a 3 to 13 times less edits made for the AB kits. So that is where I will conclude the presentation. Thank you very much for your time and your attention.
Thank you to all of our speakers. We have Stephanie, Dave, and Jaime here standing by, ready to take some questions from the audience. So as a reminder, you can submit your questions using the Q&A box on your left. Okay, so it looks like the first question is for Stephanie. Stephanie, what kind of data cleanup is required for sample DNA degradation?
Yes, so for the cleanup of the data, basically you're going to have to go and open up the raw data files inside of GeneMarker. That would be standard as far as data review from any analyst in a standard lab. And you'll go through and resolve the flags or uncertain allele calls within the system. As Jaime talked about, a lot of those might be, pull-up artifacts that you might need to clean up. Or more commonly, you will have like heterozygous imbalances or inconclusive homozygous calls that just need to either be confirmed or ignored for the purposes of the comparison that you're actually going to do.
Great, thank you. The next question is for Dave. Dave, how did the new task-based training contribute to your productivity increase?
Yeah, the new task-based training for us meant that we could take analysts who would ordinarily have had to take several months, if not longer, to get into the workflow and make a real contribution and a difference to our productivity. And it let us focus on the areas that we had real bottlenecks in, real needs in, and maybe even new technology introduced in, and we wanted to put people straight into those areas so that we could kind of relieve our bottlenecks there. And so for a couple of reasons, it made a huge impact. One, certainly new analysts who are coming into the process are useful more quickly than they would have ordinarily been. And that's encouraging on the individual level, but it also made a big difference at the wider productivity level too. And secondly, it allowed us to implement more of what we wanted to do more quickly than waiting on everyone being trained through the entire process.
Oh, interesting. Thank you. Stephanie, this is a kind of a two-part question. Did you have to perform special protocols offline before processing the samples? And how did the non-bone sample perform with the rapid hit ID? Did you have to do any special pre-processing steps? What did the results look like?
Okay, so as far as the processing before sampling, so at the autopsy level, essentially what we had them do was target the best portion, and most of the time that was the deepest portion of the tissue or the bone to get the most clean sample. If the sample that was acquired was relatively viable tissue, as well as slightly hydrated, and especially if you can see any red or blood in the sample, you can just go ahead and directly apply that sample. Then if it's either, like in this case where we had the burnt samples or really dried out or anything like that, doing that pre-wash that I discussed in the presentation. So it was just adding about 500 microliters to an Eppendorf tube, doing a quick wash, hand canting that, and going ahead and loading it after that. That helped to both clean the sample and rehydrate the sample so it could be a little bit more available for the processing. And then as similar with the brain tissue that we saw, if it was really dirty, it was just a matter of really targeting the cleanest sample that we could find. To go on to the next one, the non-bone samples performance, it worked really well. I think the highest variability that we saw was especially those highly degraded muscles. But for the most part, again, we had 28 different samples that were really wide ranging on the degradation level that we had. And all of them gave at least enough allele calls to be able to go ahead and make that comparison with our N of 7 individuals to choose from. And I think I already covered the rest of that other question.
Okay, great, thank you. Jaime, question for you. Do most labs see both pure dye and instrument-specific pull-up?
Yeah, so I think when you're talking about pure dye, that would be our traditional spectral pull-up that you see. And then the instrument specific, as I talked about, all depends on the movement of the fluorescent molecules across the detection window and how the data is captured on the CCD. On the 3500, absolutely. If you view your raw data and you start looking at pull-up closer in your raw data, you're definitely going to see both effects. You'll see the traditional pull-up peaks that center under the parent peak, and then you'll see the sinusoidal shape of the instrument-specific pull-up types. On the SeqStudio, it's a bit different. The optics are different, and so you don't see the same instrument-specific pull-up. So on the SeqStudio, the pull-up that you're seeing is the traditional spectral pull-up that we're used to.
Great, thank you. Dave, question for you. Have you had any problems with having multiple scientists on a single case, with prosecutors or defense wanting to have everyone who has touched the case testify?
Great question, and of course it makes attorneys very nervous to see that lots of hands have been all over a case. But the reality is that, sure, anybody can be called to testify if your name is on a case packet in any way, shape or form, and that's just the system we work in, and I fully appreciate and understand that. But really the person who's writing their opinions and the reports are those who are generally called to testify to what the results mean and what matters at the end of that process. And so I think what happens generally is that, at the very start of initiating a process like this, where you've got multiple analysts really in a technical sense through the lab touching a case, maybe in a batch processing system, that one or two attorneys might call on all of the analysts in the case to come testify or at least be available, but that soon gets old, especially when somebody in the middle of the process wrote on tubes or loaded the pump plate, and what does it really mean? Well, it might mean something if there's something to find, but generally it means that you just put a plate on an instrument, or that you set up an extraction. But the real important part for the court purposes is to render that opinion after you've made your interpretation on the downstream results, and so that's what we're seeing in general.
Great, thank you. Does anyone else want to add to that?
Quickly, so we have a similar system with our outsourcing lab, so not internal, but when we send samples out. And for them, they've actually done some different remote testimonies for chain of custody purposes for the larger workflows, and then had just, as Dave was talking about, the primary analyst that's signing the conclusions be the one that actually does the testimony. So, we've seen kind of similar to where Dave has, and, it might have those people needing to be available, but there might be some, lower level needs that they can do. I think ours was done with a chain of custody hearing beforehand.
And Stephanie, a question, what is the size of your agency?
So we're approximately 140 sworn officers. So we're not one of the giant big ones. We're similar to Ben Salem, if you guys listen to the panel discussion. So not one of the big giant departments, but also not super small.
Okay. Jaime, question for you. If you weren't using marker to marker on SeqStudio, what would pull-up look like with just autospectral?
Okay, so yep, marker to marker correction is the second level of pull-up reduction on the SeqStudio, and it's specific for applied biosystems chemistry only. So in the presentation I gave, the data you saw for the Promega and QIAGEN kits, that is with autospectral only, but no marker to marker. So for the kits and samples we tested, it was a 13% reduction in pull-up compared to the global filer IQC to the kits that just had autospectral. And when comparing to NGM Detect, there was a 4 times reduction for the NGM Detect kit using marker to marker compared to the other kits. So a definite improvement with the benefit of marker to marker correction.
Yeah, okay, thank you. So Dave, do you have a goal for average turnaround time for violent crime processing?
Yeah, our goal is to get it done as quickly as possible, and that is really important to us. But we have a mandate in our state for sexual assault turnaround times, like most states do now, and we want to far exceed that. And when I say exceed, I mean come underneath it. So our goal for those are 120 days, but we want to quarter the turnaround time of that expectation and include all of our case requests in that time frame. So we're really hoping and driving towards a month to 6 weeks for everything that comes in our lab.
Yeah. There was an additional question of how difficult was it for you to upgrade chemistry, equipment, and software all at the same time?
Boy, how long do you have? It's biting off a large part of that is really difficult, to be honest. It's difficult on everybody involved, just the logistics of it, partnering with folks at Thermo Fisher Scientific, just getting that timing right as well, making sure that we have the availability in the lab, and then even just the motivation and the brain power to be able to gather all of that information together and make it sensible and worthwhile. It was a lot. It's probably doing all of that together was really pushing that boundary, but I think it was worthwhile doing it rather than doing it in little bits and pieces over the course of a number of years, getting it all done and striking all while the iron's hot, I think is going to prove to be fruitful in the end.
Great. Okay. Well, I think we're at about time. I would like to thank all of our presenters today, and thank you to the audience for all of your questions. All of these presentations you've heard today will be available for on-demand viewing until June of 2021. So please be sure to share them with any colleagues who might be interested in any of the topics covered in today's sessions. As a reminder, the technical Ask the Expert chat room is now open, so head over there to participate in the discussions. I'm sure you won't want to miss that. And also be sure to visit the applications, exhibits, and poster halls for a wealth of content and to connect with Thermo Fisher representatives. Thank you for attending HIDS Virtual, and we'll see you at HIDS 2021.
Thank you.